synthesis of acetylcholine. But before synthesis of acetylcholine, what is the difference between neuronal transmission and neuronal conduction?
Conduction is the neuron stimulated. Transmission is the acetylcholine
Anyone else? Anyone else? Alishba, Mehran. There is a very clear difference between conduction in neuron and transmission in neuron. For example, this is one neuron and this is the postsynaptic neuron, right? And I have told you that every neuron have microtubules or intermediate tubules in their center, axon, right? And here we have receptors and here we have vesicles, right? When this neuron is stimulated, the sodium goes inside and potassium goes outside. That means this neuron is depolarizing. And when these signals are transferring from inside the axon from the cell body to axon terminal, this is called conduction. This is called conduction. And same neuron, when the signals are conducting from the cell body to axon terminal, is called conduction. And when these signals are transferring to other neurons in the form of neurotransmitter conduction, this is called transmission, right? This is the difference between conduction and transmission. So by cholinergic drugs, and sometimes we blocks or sometimes we enhance the conduction of neuron, and sometimes we block or enhance the transmission of neuron, right? So first of all, I have told you that the cholinergic neurons are those neurons which are working upon acetylcholine, right? What are acetylcholine? Acetylcholine is a macromolecule, or this is a neurotransmitter forming in neurons and neuronal terminals, right? And the receptors by which acetylcholines are acting are called cholinergic receptors. First of all, we will discuss acetylcholine synthesis, and then we will discuss cholinergic receptors. And when you discuss cholinergic receptors, you will easily understand the cholinergic drugs and the cholinergic anticholinergic drugs, right? So the the neurons by which acetylcholine are working, they are presynaptic parasympathetic neurons, the ganglionic and the postganglionic neurons, also the effector sites of the parasympathetic neurons. But in sympathetic neurons, only the presynaptic neurons are working on acetylcholine, and in effector side they are adrenergic, right? Because they are working upon adrenergic or beta 1, beta 2, alpha 1, alpha 2 receptors, right? So first of all, for example, I have, this is the central nervous system. I think we have discussed this a little bit in neuromuscular junction, right? So for example, from here, this is for example the nucleus, and from here neuron is arising. And for example, I am taking this neuron here. This is the cell body of neuron. This is the axon, and for example, this is the axon terminal. Right? We are discussing the synthesis of acetylcholine. Acetylcholine have how many parts? Two. One is acetate and second is choline. What is it? Choline? Choline is B vitamin, and we are taking the choline in diet. When we are eating, for example, we are eating eggs, and eggs there is a lot of cholines. And when in green vegetables we have a lot of cholines, and meat we have choline. We are taking lot of choline in diet, and that cholines are absorbing by these neurons, and in neurons they are converting into acetylcholine. But here, for example, this cell body is present in brain, right? Some of the proteins are synthesizing in cell bodies, but acetylcholine is not synthesizing in cell bodies. Only the precursor or only the vesicles in which the acetylcholine will enclosed are synthesizing in cell bodies, because this cell body have nucleus. This is cell body of cholinergic neuron. This is cholinergic neuron, right? And here we have DNA. And upon stimulation, when this neuron is stimulated, some of the genes which are responsible for the formation of the vesicles, not acetylcholine, for neurotransmitter vesicles, they are expressing. For example, here we have these genes, they express into messenger RNA, and in cell body we have a lot of ribosomes. This is, for example, ribosome, and this ribosome forms or translate this messenger RNA into a special types of proteins from which the vesicles are formed. These are, for example, neurotransmitter vesicle, but these neurotransmitter vesicles are empty, right? And two very important proteins attached to this vesicles are V snares, vesicular snares, synaptobrevin and syntaxin. They are attached to these vesicles, and these proteins are formed by the expression of the genes in cell body, right? This vesicle is empty. Now in this cell body we have a lot of mitochondria, and what is the role of mitochondria? Krebs cycle taking place in mitochondria, and when Krebs cycle is starting, we have a precursor made called acetyl coenzyme A, which is forming from pyruvate, and pyruvate is forming from glucose. When glucose is break down into 3 carbon compound, they are called pyruvate, and when pyruvate is entering by pyruvate dehydrogenase enzyme into mitochondria, they convert into acetyl coenzyme A, which is 2 carbon compound, and 1 carbon is eliminated from this pyruvate, right? So this mitochondria is present in cell body, and of course in axon we have intermediate tubules. Here we have intermediate tubules. This is, for example, intermediate tubule, and on intermediate tubules we have two very important proteins. What are the names of these proteins? They are transfer protein, dynein and kinesin. And these dyneins are responsible for taking the things from cell body to axon terminal, and this kinesin is responsible for taking the things, materials from the axon terminal into cell body, right? So they have two sides. In one hand it take this vesicle, and in another hand it takes, because mitochondria is needed here, right? And when this neuron is stimulated, what is mean by stimulation? Depolarization. Sodium goes inside, and here threshold potential take place, and at last we have minus 55 or plus 10 millivolts current occurs here at rise, and when it depolarized, this protein start walking towards the axon terminal, and this mitochondria, this is, for example, mitochondria, this is now present in terminal, and also the vesicle is present in this axon terminal, right? Here we have a very special transporter which is called calcium transporter, and this is voltage gated calcium transporter. How many types of calcium reabsorption occur in body? or absorption of calcium occur in body? Anyone? I have discussed this in very big detail in your first year.
Two types.
Two types. One is in muscle, one is in nephron. There are two very special or very dominant reabsorption occur of calcium. One is hormonal independent calcium reabsorption, and one is hormonal dependent calcium reabsorption. Which hormone is responsible for? Parathyroid hormone. Parathyroid hormone is responsible for taking the calcium from urine into blood, right? And one is paracellular transport, which occur in the response of potassium, because potassium is positively charged, and when potassium gives this positivity to the nephron or any other place in body, it repel, because calcium is also positive charge, it repel the calcium paracellularly. Paracellularly mean, for example, these are two cells and calcium is present here, and here we have, for example, this is blood, and this blood needs calcium. This cell throw this potassium into the calcium side, and this area become very positive, and when this area become very positive, it will repel the calcium in between these spaces into interstitium and blood. This is paracellular transport of calcium, and one is parathyroid, which occur transcellular transport. For example, we have receptor 7 pass receptor here, this 7 pass receptor, and for example here calcium level is low. Calcium level is low. This low calcium level stimulates the parathyroid gland, and parathyroid gland release parathyroid hormone, and this parathyroid hormone occurs as a ligand at attached to 7 pass receptor, and here the it convert the ATP into CMP, and this CMP activates phosphokinase A. And what is the function of this phosphokinase A? The phosphorylation of calcium channels. I have discussed this topic. And when this calcium is phosphorylated, the calcium from here goes to blood transcellularly. This is paracellular transport and this is transcellular transport. One transport is here, calcium, but this is ligand binded channels. These are ligand binded channels, and these are voltage gated channels. So when this voltage is provided to this calcium channel, the calcium goes inside, and when calcium goes inside, it will stimulate another receptor here, which is very important, called choline receptor or choline transporter, right? Here, for example, this is our body blood, and in nutrition we are taking a lot of cholines. Here, for example, we have choline. This choline is a type of B vitamin, vitamin B, right? And by activating this choline transporter, it takes this choline into neuronal terminal. So choline is now present in axon terminal, right? And here we have a lot of acetyl coenzyme A. It releases the acetyl into the axon terminal. Acetyl coenzyme A, but coenzyme A again leaves this terminal, right? And in response of these cholines, it release sodium outside, right? Because when the cell is taking something from outside, it also giving something to outside, right? So these choline and acetyl, they react by which enzyme? Which enzyme is responsible for catalyzing? Acetyl choline transferases. Acetylcholine transferases is working here, and it binds the choline to acetate or acetyl and formed this compound, which is called acetylcholine. And this acetylcholine is now what happened here. These vesicles have a lot of choline channels, but these choline channels are proton dependent. This vesicle taking proton from outside, right, and it gives the acetylcholine into the cell when this vesicle gives proton outside, right. For example, this vesicle have a lot of, from where these protons occurs. We are discussing acetylcholine, so it must that you know the details behind this potassium or these hydrogen ions, right, because hydrogen ion is very responsible for in decreasing pH, increasing acidity. From where this proton occur?
Depolarization.
And depolarization, protons. Proton बदल के बदा और कई गोरे, हाँ. For example, here we have principal cells in, these are principal cell. What is the location of principal cell? Anyone? Or not them? It is intercalated cell. Intercalated cell. What is intercalated cell? This is the cell between two principal cells present in the collecting ducts of kidney. Right here, one very special reaction take place between carbon dioxide and H2O, and it forms H2CO3, carbonic acid, in the presence of carbonic anhydrase. But this carbonic acid is very unstable, and it break down into hydrogen ion and HCO3 minus, right. It throws this hydrogen ion into blood, and blood takes these hydrogen ion and transfer it to the needy places in body. So for example, this is the needy place, so it transfer the hydrogen ion from blood, and this hydrogen ion well goes into the vesicle, and in response of this hydrogen ion, it will take, for example, this hydrogen ion goes outside and it takes acetylcholines inside. This is the transport mainly, right. What will happen to this hydrogen ion? This hydrogen ion will again retake by blood, and in blood it will secrete into the urine, and in urine it will react with the ammonia and will forms ammonium, which is less toxic, right. So this is the transport. So now we have a lot of choline in the vesicle. For example, these are a lot of acetylcholine, and these acetylcholines are responsible for activation of cholinergic receptors, right. These vesicles have a very special type of proteins, and these proteins are called V snare protein, synaptobrevin. They are called synaptobrevin, right. And these terminals have also very special types of, first of all, for example, I have this binding site. This is the binding site, but this binding site is blocked by V snares, and this vesicle is not able to attach to any other proteins. And this terminal have, for example, the receptor to which this arrow can bind, but this site is also blocked by T snares are syntaxins, right? Now we have a lot of calcium channels in these vesicle. For example, this is calcium channel and these are calcium channels, right? From outside it will take calcium. These are voltage gated calcium channels. So this voltage is enough for activating these channels. And for example, from here also calcium goes inside. When this calcium goes and attached to this snare and also attached to this snare, right? So for example, if we have these two proteins and here is also positive charge, here also positive charge, it will repel each other, right? And when it repelled, this site will become like but they have attached calcium. Calcium is responsible for repelling these sites and now this binding site is open. This calcium is also came here to these syntaxins and they are also repelled from each other. So this site is also now open, right? So for example, I have these activated channels here. Right, and they have now calcium attached and this site is free to, for example, this is the binding site of the vesicle. Right, now they are both are attached to each other and it contracts the vesicle, and when this vesicle is contracted it will release the acetylcholine into the neuromuscular junction or neuro junction, right? For example, I have this is a muscle cell, right? And we have nicotinic receptor here. What are nicotinic receptor? They are cholinergic because they are activating by choline and they have ligand binding sites because these channels are ligand gated ions channels and these channels are voltage gated ion channels. So it have a site for acetylcholine, and when these acetylcholine are released, for example, these are acetylcholine. When this acetylcholine came here, it will attach to this receptor and it will activate this receptor, and when this receptor is activated the sodium will goes inside, and when sodium is goes inside this cell will go into excitatory postsynaptic potential, EPSPs, excitatory postsynaptic potentials, right? And by this potential it will activate the other voltage gated sodium channels and all this area is polarized, right? And this structure here is called triad. This is called triad. Why it is called triad? It have two sarcoplasmic reticulum and one T-tubule, transverse tubule. Collectively it is called triad and it stores a lot of calcium, right? And when it activate the pyridine proteins, hydroxypyridine proteins are this, they have sequestrine proteins, it opens the channels into the cytoplasm and this calcium goes outside and here contraction of muscles occurs, contraction of muscles, right? This is the neuromuscular junctions also, and this is the action of acetylcholine and this is the synthesis of acetylcholine. But this acetylcholine is not an unimportant thing. This is very important in body. So it will be separated into its parts, the choline and acetyl, and will reuptake by this terminals, right? So for example, we have 12 pass receptor here. What are 12 pass receptor? Reuptake channels, reuptake receptors. First of all, we have here, we have, for example, this is the acetyl part of acetylcholine, and this is the choline part of acetylcholine, right? And we have here a very special enzyme called choline esterase. Choline esterase enzyme. For example, here we have this is choline esterase enzyme, and this choline esterase enzyme separate these two into choline and acetyl. This acetyl is again goes to the cell and working, and this choline is transported to the exon terminal again and will recycle by acetate into acetylcholine, right? So this is generally the synthesis of acetylcholine. Now, which drug can affect the acetylcholine transporter? The calcium receptors or the nicotinic receptor or cholinergic receptors. Generally, we have one experimental drug which is called hemicolonium. Hemicolonium. For example, I have choline transporter here. This is choline transporter, and we are taking hemicolonium. This helium occurs and blocks this choline transporter, and now this exon terminal is not able to take choline. For example, this is now blocked and choline cannot goes into the exon terminal. When choline is not able to go into the exon terminal, the acetylcholine will not formed here, and when acetylcholine is not formed here, it will not able to activate these receptors, and when these receptors are not activated, the contraction will not take place. This is one example, contraction, right? Dilation, a lot of examples are of nicotinic receptors, right? The next drug which is responsible for blocking these receptors, acetylcholine transporter. For example, I have this vesicle here. This is, for example, vesicle for acetylcholine, and this acetylcholine is trying to go into this vesicle by which transporter? Acetylcholine transporter. We have one another experimental drug trying by the scientists trying these drugs on animals, right, which is called visamicol. This drug is called on rats and rabbits. Visamicol. This visamicol drug blocks acetylcholine transporter, and now the acetylcholine is not able to go into the vesicle, and when acetylcholine is not going to vesicle, it cannot exocytose to synapses, right, and not able to activate the nicotinic receptor in postsynaptic neuron. This is the another drug which can affect the neuromuscular junction or acetylcholine, right. The next is one toxic substance which can produce in the canes. The foods which are stored in canes are sometimes they are toxic, right, by which the Clostridium botulinum bacteria, it is responsible for formation of botulinum toxins, and botulinum toxins are responsible for cutting the T snares and V snares. They have knives, right. For example, For example, here I have botulinum toxin. Bottle. Remember when you said bottle, canes, right? For example, we are giving the honey to children. They are stored in cans. We are eating cans food, and sometimes the bacteria produce the botulinum toxin and dead cans. And when we are taking the cans or the food, it will perform these actions. We have botulinum toxin, and this botulinum toxin is produced by clostridium botulinum bacteria, and clostridium botulinum bacteria is grow in canes, right? For example, this bacteria came here, these toxins came here, and they have a very special knife, right? These are botulinum toxins, and these botulinum toxins are responsible for cutting T snares and V snares. And when T snares and V snares are cutted, this vesicle is not able to attach to these receptors, and when it is not attached to these receptors, the acetylcholine cannot come into these synapse, and these pathways are not activated, right? The next, you know, spider. What is spider? झोला पे जाने झोला दागे की ओके सम टॉक्सिन्स दी पर झोला के तोड़ झोला खलक यादी कना वो जदी चक खतरनाक की दागे चक कोले खतरनाक की द ब्लैक स्पाइडर ब्लैक विडो स्पाइडर व्हाट इज द नेम ऑफ द ब्लैक विडो spider, right? For example, here is the black widow spider, and it produces some toxins called, they are called laterotoxin, laterotoxins, and this laterotoxin and botulinum toxins are working against each other. For example, this laterotoxins came into the axon terminal, and when it came into axon terminal, it will binds more acetylcholine vesicle to these receptors, and we have a lot of a very high amount of acetylcholine is now present in the synapses due to the laterotoxins, and when these laterotoxins affect this synapse, it cause the cholinergic crisis. Cholinergic crisis mean poor stimulation of cholinergic receptors by laterotoxins, and it cause cholinergic crisis. There is overstimulation occur, over contraction will take place, over sweatening will take place, right? So these are the functions of some drugs on neuromuscular junctions and acetylcholine. The last one is the tuberculosis drug. Nowadays it is also used in operation theaters for paralyzing the muscles, right? In past eras, when someone wants to paralyze a person, they take, for example, this is the arrow, they will take the tuberculosis drug and will apply on this arrow, right? And when this arrow hit a person, it blocks these receptors. For example, it blocks these nicotinic receptors which are present on the postsynaptic neurons, right? This is tuberculosis drug. Tuberculosis drug, right? Where it affects, for example, this is the presynaptic neuron and this is the postsynaptic neuron or neuromuscular junction end plate, and here we have a nicotinic receptor. These tuberculosis drug blocks these receptors, right? And when these receptors are blocked, the acetylcholine is not able to activate these receptor, and paralysis occur in these muscles, right? So for example, if these are the tuberculosis drugs, right, and these acetylcholines are not able to activate this receptor, this contraction will be inhibited and the muscle will remain in relaxed position in paralytic stage, right? So these are the effects of some drugs on the neuromuscular junction, and this is the formation of acetylcholine. And acetylcholine synthesis, first of all we need choline, and this choline is coming from nutrition. We need acetate, which is coming from mitochondria. We need vesicles, which is coming from ribosome or cell body. Also we need proteins, which are attached to the vesicles, coming from cell body, right? And when these acetylcholines, we need choline transporter present on axon terminals. Also we need acetylcholine transferases, which are present in axon terminal, right? Now acetylcholine is formed. Choline plus acetyl, it forms acetylcholine. This is the synthesis of acetylcholine and enclosed in vesicle, right? For action we need nicotinic receptor, which are present on the postsynaptic end plate or postsynaptic neurons, right? And now we will discuss, this is the main thing which is responsible for the function of acetylcholine. There is one disease which is called myasthenia gravis. Myasthenia gravis. What is myasthenia gravis? Anyone? Hmm? Good. When these tuberculosis drug occur here and it blocks the nicotinic receptors, now acetylcholine cannot working, and this condition is called myasthenia gravis. Also, when these calcium channels are affected, and this calcium is not going to inside. When these calcium are not going to inside, it means that these vesicles are not able to attach to these receptors and the muscles will be affected. This is called myasthenia gravis, but myasthenia gravis have two types. It have two types. One is called Lambert-Eaton syndrome, what is called myasthenia simple, myasthenia gravis. Myasthenia gravis and generally there are antibodies produced against the calcium channels of axon terminals, antibodies, and due to formation of these antibodies the calcium is not able to go into the axon terminal, and these acetylcholine vesicles are not able to attach to the terminal, right? This is called myasthenia gravis. Eaton-Lambert and Eaton-Lambert syndrome, how you will diagnose the Eaton-Lambert syndrome and myasthenia gravis? Why it is called Eaton-Lambert syndrome or Lambert-Eaton syndrome? Because when these channels are inhibited, a lot of acetylcholines are not able to activate these receptor, and when this person when he is working, he will get fatigue very early, very soon he will get fatigue. And when these calcium channels are blocked, and this person working for a while, he will activate some of other calcium, because we have a lot of calcium channels, and when this calcium channels goes inside the axon terminal, it will activate these receptor, and with time he will be stimulated. This is called Lambert-Eaton syndrome. For example, myasthenia gravis दो types थी. Now we have cholinergic receptors. Cholinergic receptors are now very easy. We have two types of cholinergic receptors. One is nicotinic receptor and one is muscarinic receptors. Right? Dauran, come on.